All right, so we have Christine. She's a PhD candidate, Christine Hustmyer, right? Is that how you?
Yes, that's right. Yeah.
Okay, so Christine is going to talk about bacterial genetics. And one more rule, you guys, after I'm going to lock the meeting out at 5. So if folks, all your classmates that want to try to be late and only sit in for 15 minutes or so, they're going to get locked out and won't get the credit. You guys will get the entire thing. So if they intend to get on, better call them. But with that, here's Christine, and she's going to talk about bacterial genetics, and she's going to tell you all about herself and what she's going to discuss today. So welcome, Christine.
Thank you. Thank you so much for having me today. It's really great to see you all here on a Friday. And I'm really excited today to talk about what I do as a graduate student. So I'm still in school just like all of you folks. I've been in school for a while, looking to get my PhD coming up soon. But what I do is I study bacteria and the different ways that bacteria behave in certain environments and certain disease states. So I'm going to show you a little bit about some of the tools that we use to monitor how bacteria are behaving. So this is an example, hopefully you folks can all see this slide here, of what we'll talk about today, which is this glowing bacteria. And how we as scientists can understand how the bacteria are behaving by manipulating them in certain ways or by performing different genetic mutations to the bacteria. But first, I am all the way in Wisconsin, so I'm a little bit further from you guys in California. This is Madison, Wisconsin, at the University of Wisconsin. It's a really great institution to be at. We have lots of life sciences programs, encouraging people from all different backgrounds to join and participate in different STEM fields. So this is the campus here at the University of Wisconsin, and my lab's right over here. And we sit on this really small body of water, this called Lake Monona. And so this is the view from my lab, which that's where I'm at now. This is our lab. And so I have a lake view, which is nice. And then this is my bench where I do most of my experiments. So I have all sorts of different reagents, like different liquids that I mix together. I have my notebook to record different observations. And I have all sorts of different bacteria that are growing at my bench and in different incubators across the lab. So mainly today, I'm going to talk to you all about a bacteria called E. coli, which some of you may have heard of, or you may have gotten sick from it if you've eaten any sort of contaminated meat, or you might have heard about it in the news. And that's really what I study is I try to understand how E. coli does certain behaviors and when it does that, with the idea that if we understand how the bacteria are working, we can help prevent certain humans from having diseases that are caused by the bacteria. Okay, so the crux of my research is sort of the central dogma, where all behaviors of certain bacteria or of humans, sort of universal, occur from different proteins. These proteins are sort of expressed or turned on or off in certain environments and certain states. And it's our job as scientists to understand what these proteins are doing. And so what I do specifically as a bacterial geneticist is I'm trying to focus on how the DNA influences what proteins are turned on and subsequently what bacterial behaviors are turned on. So the key really is in this DNA sequence, which I'm sure many of you all have learned about, right? But DNA is really the key to understanding how different bacterial behaviors are turned on or off. And so you might think, well, bacteria are really small, right? They should be much simpler compared to humans. We're much bigger. We have a lot more DNA compared to bacteria, but we really still have a lot of open questions about bacteria and understanding how they're causing disease in humans. And that's really where my role comes in as a researcher. And that's what I'm doing my PhD on. So what I specifically study is this bacteria that causes urinary tract infections. So Maybe some of you have heard of those diseases, or maybe you've had your parents might have had a urinary tract infection, or your grandparents. It's a really common infection all across the globe. And these infections we know are caused by this bacteria called E. coli that I also said causes food poisoning. But this E. coli that causes these urinary tract infections are really different in that they have very specific environments that they can persist in, right? So normally E. coli that causes food poisoning might live in your meat that you eat, right? But urinary tract infection E. coli actually can live in humans and persist both in the bladder of humans and all the way up to the kidneys, into your blood. And that's really dangerous for humans, right? Because we don't want bacteria in our blood system that can cause a lot of harm. So it's my job as a bacterial geneticist to understand how this little bacteria can persist and live and thrive in all of these different environments in the human body. And more specifically, how it's using its DNA to turn bad behaviors on or off. So that's really what I do in my work. And so I need to have a way to actually monitor how this bacteria is behaving. So to do that, I turn to the bench, right? And I'm looking at this bacteria that is super small. And I often use agar plating to understand how these bacteria are behaving. So here is an example of a E. coli plate that's growing. You can see that it looks kind of gross, and it also doesn't smell very good. But this is how I am going to then understand how different bacterial behaviors are turned on. So on this plate here, this is called an agar plate. And this is actually an example here that I'm holding up. It's really interesting because on these agar plates, maybe you guys can see it here, it's sort of a solid plate. You can see that my spear here is not going through it. And these agar plates are really neat because we can then go ahead and pick a specific bacterial, what we call colony, from this plate. And within that colony, there are millions of bacterial cells that we can then study to understand their bacterial behaviors. So to understand just a little bit more about how we are isolating these bacteria, I just want to spend a little bit of time talking about what this material is made of that we actually pour these bacterial plates from. And so I mentioned that this is called agar. So agar is a basically nutrient-rich media here, or medium, that the bacteria like to grow on. So a good way to think about it is if you all have ever had Jell-O, it's kind of fun to play with, it's super jiggly. Jell-O you make by mixing a bunch of components together with some really boiling hot water, and then as it cools, it solidifies into this sort of Jell-O material. That's the same thing that agar is, right? So here with this agar, it looks solid on the top, but I can actually poke into it like so. And that allows me to plate the bacteria on the top and allow for the bacteria to form on the surface rather than being inside this sort of Jell-O material. So I can always access it from the surface, if that makes sense. So how did agar actually come about? So most scientists at the University of Wisconsin are probably using these bacterial agar plates all the time to do their experiments. But how did it actually start? And the answer is actually from female scientist Fanny Hess back in 1882. So back in the early 1800s and mid 1800s, researchers knew about bacteria. They were interested in understanding what it was doing, but they were having a really hard time working with it. So oftentimes they would use different materials to try to grow the bacteria. They were trying different temperatures to grow the bacteria, and that wasn't working out very well. So what Fanny Hess did was she capitalized on her ability to make different Jell-Os and different jellies. And what she proposed is that with this agar that they were using at the time, they were literally actually using the material that you use to make Jell-O. They were using gelatin as a component. And that didn't work well because when they would put this agar at higher temperatures that the bacteria need to grow at, this agar would completely melt. So that wasn't good, right? Because then the bacteria is mixing in with this material and it's like a sort of gross bacterial soup. And that doesn't help the scientists to understand the bacteria because they can't isolate the bacteria. So what Fanny Hess suggested was she had read and she knew from her jelly making experience that instead maybe scientists could use agar, which is isolated from seaweed. And you can see that here. It sort of has a similar jello-like material. But what's great about agar is that it doesn't melt at the temperatures that bacteria grow at. So you can still isolate this bacteria from the top of the plate. So that's really the crux of how agar came about. And this is the sort of workhorse material that most bacterial geneticists use to understand bacterial behaviors. So what do I mean sort of at the scientific level when I say bacterial behaviors? So you can think of a bacterial behavior as something that causes a disease in a human, some sort of material that is being released by the bacteria in the human to cause harm in humans. And we know that these bacterial behaviors can be studied genetically. So you folks may have also learned about in some of your classes this idea of in each of us, right, we all look different. We have different colored eyes or different colored hair, and that's known as a phenotype in humans. Phenotype is all sort of driven by genotype or the specific DNA sequence that a person has that leads to this different phenotype or this different characteristic or trait. And so what you might not know, though, is that these bacteria, the gross little particles that I showed you in an earlier slide, that these also have different phenotypes and different genotypes, right? And so even though they are sort of simpler than humans, there's a lot still to uncover about how their genotype changes into their phenotype. And so what we want to do as bacterial geneticists is we sort of do a reverse here, where we're monitoring what phenotypes occur in bacteria, and then we can hone in on what genotypes are occurring so we can prevent certain bad bacterial behaviors from occurring. Okay, so in this scenario, my big question is, how do we know when certain behaviors in bacteria are on or off? So how do we know when those different DNA sequences that I talked about, when those are on or off? If I'm just looking at a bacterial agar plate, like I showed you earlier, all I'm looking at are some gross looking bacteria. Well, what we can do is we can focus on genes that we think might cause bad bacterial behavior. And so genes are a group of DNA sequences that encode for a protein, like I showed you in that central dogma slide. But you can really just think of it as a sort of group of DNA sequences that lead to a bad bacterial behavior. So you can think behavior, certain DNA sequence. So in this scenario, right, we have this bacteria. It has some sort of bad bacterial behavior that it can turn on to cause harm in us as humans when it infects us. We want to know when this bad bacterial behavior gene or bad bacterial DNA sequence is actually turned on. And so to do that, we're going to use this special feature called GFP. Now, GFP is a glowing protein. It's a glowing feature or glowing DNA sequence that we can add and manipulate into the bacteria near a bad bacterial behavior gene. And then we can monitor when different light is occurring. Okay. So in this scenario, if I've added this GFP to this bad bacterial behavior, I can monitor if this bad bacterial behavior is turned on when this GFP is making this glowing behavior of the bacteria. So what does this look like in principle? Okay, so I showed you folks these plates earlier. So we have these really gross looking E. coli. They're yellow here, like so. And I've just drawn some pictures on them for the purpose of this class. And so right now, these two plates look the same, right? This is on this agar plate. This bacteria is growing on the surface. But I know that I've added GFP to this bacteria. And I know that if a bad bacterial behavior is turned on, then I can monitor that this behavior is turned on because I'll see this glowing phenotype or this glowing picture. And so I'm going to show you what that picture looks like now. So here is now in the dark. This GFP is really easy to see in the dark. When I can excite it at a certain light that it likes to be excited at. And now you see that compared to this plate where this bad bacterial behavior is off, you don't see much glowing. But when I have this GFP present and this bad bacterial behavior is turned on, I see this really neat green glow. So essentially what bacterial geneticists are doing is they're trying to figure out when this green glow is happening so we can better understand the timing at which these bad bacterial behaviors are turned on or off. And so now as a scientist, I could say, well, I know under this condition that this bad bacterial behavior has been turned on, and then I can try to figure out what led to that bad bacterial behavior being turned on. So what else can agar plates tell us? We can also add different sort of features to these agar plates. So this has all sorts of material that the bacteria really like to grow on. It's sort of like a very high salt diet for the bacteria. Some of you might also like a lot of salt, so do bacteria, so that's something we have in common with them. But what else can we do with agar plates to understand how bacterial behaviors are turned on or off? So what we can also do, and this is kind of gross, right? But what we can also do is we can put a little bit of blood in these agar plates. And so this is important because the E. coli that I told you about earlier that causes these really bad infections in humans can spread to the blood. And when this bacteria spreads to the blood, it can cause these bad bacterial behaviors to turn on, and it can actually lyse human red blood cells or break open human red blood cells. And that's really bad for humans, right? We don't want that to occur. And so we need to figure out how we can stop the bacteria from doing that. So the way we can monitor if this breaking up of your red blood cells or human red blood cells is occurring is we can add blood to these agar plates. And then in this scenario, what we can do is we can add the bacteria to this agar plate and it'll grow at the surface. And so you can see this white here is this bacteria, and it's actually making this factor, this bad bacterial behavior, that's lysing these red blood cells. So that's what that black ring is here. It's breaking up those red blood cells or killing them, and it is a better way to say it. That's again not what we want to occur in humans, right? So we need to figure out a way that we can prevent that from occurring in humans. And one way to do that is that we can change different DNA sequences in this bad bacteria to understand if that changes how much breaking of those blood cells occurs. So here what I've done is I've just compared to this normal bacteria all the way on the right. You don't have to worry about these terms here. Compared to this normal bacteria, we can do things like change a certain DNA sequence, like so here. You can see that black ring is now much smaller. So these bacteria are much worse at causing bad things to happen in humans. And so this is a great way that different chemists or biochemists could then maybe develop a different drug or a different medicine to help prevent the bad bacterial behavior from occurring in humans. And this is all through monitoring what happens on these sort of jelly little plates. Okay, so overall, what can we learn from these bacterial agar plates? So agar plates in their current form were devised originally in 1882 by female scientist Fanny Hess, and it all stemmed from her love of cooking and making different jellies. So it's just to show you that you can have many hobbies and still pursue science, and oftentimes things that you learn and things that you bring in from any background can help you be a better scientist and help you think of new ways that maybe traditional or majority folks in science might not think of. So every voice is really important in the scientific field. Also, I've told you that agar plates help scientists to specifically look at one bacteria by growing on the surface. And this can help us to better understand how different bacterial behaviors cause different diseases in humans. We can monitor bad bacterial behaviors by using this glowing protein called GFP, which helps us to understand if a bad bacterial behavior is turned on by this glow. And also you can use blood auger, even though it seems kind of creepy, it's really important for us to study how these bacteria can hurt and harm our blood in order to better understand how to prevent these diseases in humans from causing us harm. And so that's overall what I have for you folks today, but I'd love to hear your questions, anything about this presentation or what life is like as a scientist, what I like and what I don't like about it. I would love to hear what you have to say.
Okay, I clicked on the chat and the unmute. So if anyone has any questions, go ahead. Let me make sure I did. All right, let's see. Okay, there we go. There's one.
Yeah, so the question was, is life as a scientist fun? I do think it's fun. So you get to work with your hands a lot. So I would say anyone who enjoys working with their hands or reading or writing, there's something for everyone in the science realm. You also get to work with a lot of nice people. So I'm here in the lab right now, and there are a lot of other scientists in the other room. So that's been really nice. Science is also sometimes a little tricky. So it's important to really be persistent and keep trying. Those are all skills I know all of you have. So, okay, how did I discover that I wanted to be a scientist or microbiologist? Yeah, so I in college, so you guys are already ahead of the game, but in college, I joined um, uh, a sort of women in STEM research group, which paired me with a mentor and gave me I had to take classes with a bunch of other female scientists, and that really helped me understand that I do really like science and being a microbiologist. And I really enjoy sort of asking questions that no one knows the answer to. It makes it sort of exciting because you're sort of the first one to know something, and that can be nice. How much do I get paid as a scientist? That is a good question. So I'm a grad student right now, so I do get paid a little bit of money, which is nice. So often in science grad schools, you're able to get a little bit of funding to go through, or a little bit of money to go through the grad school. Once you have your PhD, or there's lots of science opportunities without a PhD as well, you're generally making a good, decent amount of money, and there's a lot of need for good STEM people like all of you. Okay. How early do you wake up in the morning every day is the next question. So sometimes because the bacteria grow at different times, you want to come in when they're most ready to work with. So that can cause a little bit of early days, but I usually get into lab around 8 or 9 o'clock and usually leave around 6. So that's pretty standard, and that's what most of us do, I would say. Although there are some bacteria that take a really long time to grow. And so that you sometimes might have to come in at night to work with them. What do you not like about your job? I would say that science, because everything is unknown and you're always asking a lot of questions, sometimes it can get hard because sometimes the questions that you have planned and the experiments that you're doing don't always pan out. So you have to be really good at accepting failure. Not failure per se, but things aren't always going to work out the way you planned. There's not a lot of structure in science, and that can be hard, but it's something that you learn. And that's something you guys are all probably learning in school, right? Is how do you get back up after you've had a hard day, right? And that's a key trait for a scientist, is just being able to keep trying. And as long as you keep trying, something good will come of it. Yeah, so the first time I was in a lab like this, I was very nervous. I had a PhD, so a person like me mentoring me, and I was working with actually an auger plate like this, and I was so nervous as I was working with it. My hands were shaking and stuff like this because I was whole new and I didn't think I fit in. And I actually stabbed through the plate like so and completely ruined the plate. But my mentor was really nice and just encouraged me to keep trying. So I think everyone is a little nervous the first time they're in the lab, but things always get better as long as you keep trying. Okay, the next question is, since proteins are microscopic, isn't it hard to insert them into bacterial cells? That is an excellent question. That's from Neil. Yeah, so that was a good observation. So what we do is we're actually inserting the protein through the DNA sequence. So it's a little bit easier to manipulate. It's a much smaller area. And so with that, we can use different materials to get the DNA sequence into the bacterial genome or the bacteria DNA. We actually use something called a phage, which is a bacterial virus. So you all have probably heard of viruses. Bacteria have viruses too. And we can use that bacterial virus to sort of infect the bacteria with the DNA that we want it to have. And then we use the bacterial machinery itself to make that protein. So the bacteria itself is making the GFP. It's not us telling it to do so. But that was a really good question. So lots of different ways we can manipulate the bacteria into doing what we want, but it's normally the basis of doing that is through the DNA sequence. What tools do you use to get into science, like a microscope, for example? Yeah, so I use a lot of microscopes. I also use a lot of pipettes, which I can show you here. I'll just grab them from my bench. So this is a pipette. It helps us to precisely pick up different liquids. So we use this little knob here, and we can pick up different liquids and move DNA around in that way. We also use different ink or ovens for the bacteria. So just maybe if you guys are making a recipe with your family, it's almost Thanksgiving, right? So you might be baking a turkey or something. Bacteria also have different temperatures and incubation times or cooking times that they need for them to grow. So I use those a lot as well. Have I made any discoveries? That's a good question. Yeah. So all of us, we make discoveries, but some of them are smaller than others. So I have made a couple of discoveries about this bad bacteria that causes disease in humans. And oftentimes what we do when we have these discoveries is we write a paper and we publish those papers in what's called a scientific journal. And then other people read those papers, and then eventually, if probably not my discoveries, but some other really influential discoveries, those go into the textbooks that you all learn about in class. So that's sort of a neat way that researchers are involved in school that you folks learn eventually, although it sometimes takes some time for discoveries to really be understood as really impactful and lasting. Yeah, okay. The next question is, do you conduct research on your own or do you have a mentor? So I do have a mentor or like a teacher. So I'm still a student like you all. So I have what's called we call them a principal investigator. And my mentor has been around for a long time. He's in his 70s. He's a very good scientist. And so he has a lot of experience in helping the grad students to figure out what's the best experiment to do? Where do I go when I feel like none of my experiments are working? How do I keep going? So I do have a mentor. But then once I have my PhD, then hopefully I could start to do research on my own. Okay, do your contributions direct to discoveries or do you just do testing? Yeah, so that's a good question too. I do both. So oftentimes we do follow the traditional scientific method that I'm sure you may have been drilled into in your classes, the idea of you have a hypothesis or an idea, and then you want to test that idea with different experiments. And oftentimes what happens is you have an idea and then you test it with a bunch of experiments at the bench, and then you realize your idea was wrong. And then you have to go back and reform your idea. So it's sort of a circular pattern. Or you have an idea, you test that idea experimentally, and that idea turns out to be right. But then there's all of these other questions that then arise from your hypothesis being correct. So it's really sort of a circular motion where it almost seems like sometimes science never ends, because there's always going to be an additional question that arises from a new discovery. Yeah. What do you do in your free time? Okay, so I like to run, which is really nice here in Madison, Wisconsin, although it's getting very cold. We get a lot of snow here, so that's not as great. I'm also really into race cars, which may seem a little bit unusual, but I love auto racing, not participating in it, but watching it. And I also really like to cook and to bake. So those are things that have helped me in the PhD to still, enjoy life outside of the lab. Yeah, okay, the next question is, what are some additional examples to turn genes on and off besides GFP? That's a really neat question because we're at a really important time in science in what you would call genetic engineering or changing different genes. So one that you might have heard of is CRISPR-Cas9, which is also a sort of bacterial virus that scientists have learned to manipulate to help make specific edits to the DNA. And that's even as close to sort of an A or a T in the DNA. They can switch a one nucleotide to change how a gene is turned on or off. So that's a really neat feat that scientists have developed. I think there's a lot of questions about when we're doing these bacterial genetics, right? That seems fine because they're bacteria, but once you start turning on and off genes in humans, right, how does that sort of get monitored? And that's something to think about ethically long term. Great. The next question is, what was the first bacteria that I worked with? So the first bacteria that I worked with was called Vibrio harvii. And that is a bacteria that infects shrimp. Many of you might like shrimp. It causes a lot of death to shrimp and therefore is a big bacterial burden in the agriculture industry. And that bacteria is really neat because it naturally bioluminesces. Here, I might look up a picture of it just a moment. Okay, just gonna share my screen. Oops. Okay, so maybe you guys have seen these like nice pictures of these jellyfish or this bioluminescent ocean. This is caused by that Vibrio bacteria. And these bacteria glow when they are in this group. So they can actually talk to each other. And then they form this beautiful blue color once they're in a group. So it's kind of a neat thing to work with, right? Because they're working together as a team to do a different bacterial behavior. Okay. Okay, next question is body itchiness related to bacteria or is it something else? I think that it's oftentimes something else, but I could be wrong. So there could be, if for instance, if you have like a wound or a cut and that itches, that could be a bacteria infection into that wound. But oftentimes that might be something more related to human behaviors or different genes that are being turned on or off in humans. Okay. So Charity asked, do you develop things for medical science? So that's a neat question too. So I am in the College of Agriculture, College of Farming, essentially here in Wisconsin. We're in the middle of the United States. There's a lot of farming going on. So most of my research is more traditional in terms of what we call basic science. So how do we just understand interesting questions in general that don't always have to have medical applications? But basic science can always lead to really important medical breakthroughs. Like for instance, the COVID vaccine, right? That was all developed from basic science, people trying to study different properties about life that then led to these different medical applications. So yeah, that's that question. Okay, so Cameron, these are all very good questions, guys. So Cameron asked, did you love science and do science activities as a kid? That's neat. So I did do some science activities as a kid. I really liked building Legos. So I don't know if any of you guys are Lego fans. I enjoyed that and I participated in some science fairs. But it really wasn't until college or so that I started really liking science. So I would say that that if you like science now, that's great, but you still have a lot of opportunity to explore different areas to find out what you want to do. And it's never too late to get into science. It's great that you all are interested in it now, but there's plenty of opportunity down the road. Okay, Ethan asked, what was my favorite experiment? So I like those blood experiments because they're very striking in terms of you get a really clear result as to this seems like a really great way we could help prevent this bad bacterial behavior from occurring. I also do a lot of microscopy experiments where we can freeze the bacteria and we can get different pictures of the bacteria and how they're behaving. So those are neat too. Okay. Okay, next question is what college did I go to? So I am from Indiana, again, if you guys can't hear my accent, the middle of the United States. And I went to Indiana University, and that's where I started working in a lab. So it wasn't until college. And even you can do it later in life too. We need everybody in science, but that was where I went to college. Our colors are red and crimson. Okay, did you ever see yourself, Cameron asked, did you ever see yourself becoming a science teacher or having a science related job in the future? Yeah, so I'm really interested in teaching science. And so that's part of the reason why I'm getting my PhD is I'm really interested in teaching science at what we would call a community college or a college for primarily college kids. And so that's one of the main reasons I got into science. I had a lot of great science teachers growing up who encouraged me, like many of your science teachers are encouraging you to. Is red tide a type of bacteria that is similar to bioluminescent bacteria? That is a good question. I'm going to look that up. See, I'm always learning something new. I'm learning things from you folks. Okay, I think red tide is an algae, which I think is a fungus. Yeah. So I believe that red tide, which also sort of makes this glowing color, that's a fungus or a yeast. So they're related to bacteria, but they have more DNA sequences and more behavior. So they would be classified as sort of more complicated than what I study. So that was a good question too. Okay, Ricardo asked, have you ever had an accident with the bacteria? That is a good question too. So these bacteria, some of them are really safe to work with because they've been used in the lab many, many years over. And so we have a lot of safety procedures to work with them. But some of them, like that bacteria that causes those really bad urinary tract infections, those are really dangerous. So we have to be really careful not to accidentally sort of stab ourselves. Or sort of needle ourselves, for instance, because then we can immediately get a really bad bacterial infection. So I haven't had a ton of accidents with the bacteria, but I would say that some people, there's a lot of lab mishaps. Like we have open flames going sometimes, which sounds kind of scary, but like think of it as like a huge candle that's going on. And this open flame helps keep the environment really sterile so the bacteria can't grow. But a couple times in lab, that open flame has caught onto some paper that's hanging around and caught that on fire. So that's definitely been not a good thing to happen in the lab. Do you have a favorite inventor or a favorite scientist? Cameron asks. I do. So my favorite scientist is Marie Curie, who won a couple Nobel Prizes. She discovered, basically is the mother of radioactivity. And that's something that we use all the time in the lab to look at different DNA molecules. And so she's my favorite scientist. She, unfortunately, at the time, they didn't know that radioactivity was really harmful. So she passed away doing her science, which obviously is awful, but she was a really neat person, especially to be a female scientist when a lot of female scientists weren't accepted. So that's one of my favorite scientists. Okay, Isabel asked, so I have a question about being between biology, health sciences, and microbiology as a major for college because this person wants to go into the medical field. So the question is, do you have any advice related to microbiology majors or fields if there's suggestions? So I think that, so I have a degree in biochemistry and biology, which are pretty general, but I obviously really like bacteria. So I would say if you really like bacteria, then I would definitely go for a microbiology degree if you know that that's what you want to do, or the study of bacteria. But it also doesn't, I think, having a more broad survey, which is what you would get from a biology degree, that'll also help you. So anything will serve you well. I think just following what you like versus what you think might be good for getting a job is always sort of the best advice. So just seeing where your heart is taking you and picking a science degree from that is what I would do. Did you have a specific teacher that inspired you to pursue a career in teaching? Yeah, so I had a lot of great teachers really in middle school. So I know many of you all are in middle school, and I'm sure you all have your favorite teachers. Maybe some are here who have helped you along the way and made you really like a subject. So yeah, I've had a couple of teachers like that. A lot of my teachers are actually history teachers, even though I'm in science. But I think it's all about just enjoying learning, and that's really key for being a good scientist. And so if you like school, you'll be a great scientist. Okay, so the next question is, what are the origins of bacteria? So bacteria have been around for a really long time. So probably before, well, we know before humans. So there's bacteria that are found in different planets that aren't found on Earth, probably in different atmospheres as well. So because they're so simple and because they're so small, we think that most of life probably originated from different bacteria at some point in time. So that's where they're from. So actually in the lab right next to us, we have people who are studying different bacteria on different planets to understand what the first bacteria actually was. So that's sort of interesting. Cameron asked, have I ever watched Bill Nye the Science Guy? Yes, I have. I have watched Bill Nye the Science Guy. I enjoy Bill Nye the Science Guy a lot. I think that I hope you guys like him too. It sort of shows my age that we maybe have all watched that, but I do really like Bill Nye the Science Guy. Have I watched, the next question is, have I watched The Magic School Bus? And yes, I have. Yeah, that's also a great episode, or a great show. I don't know, have you guys seen the episode where the school bus like goes into the stomach of a human and stuff, and it's going through the different, yeah. That one's a good one. That one's really neat. It helps you really think about science in a different way. You guys have any other questions for me? No? Okay. Well, you all can read here. I'll just share my screen one more time. I just want to tell you that if you ever have any questions or you want to talk one-on-one about, if you're interested in going to grad school or thinking about, I know you all are from different backgrounds and you're at different points, but if you're ever interested, you can email me if you want to take a picture of this. This is my email address, and then I'm also on Twitter, or X, I guess is what it's called now, if you need to contact me.
Or just contact us or ask Mr. T.
Yeah, there we
You
go.
can contact us and we'll find you.
Great.
So if you guys don't have any more questions, you guys can have your Friday evening back. Thank you, because I know this was a holiday and school was out today. So we appreciate you guys taking the time and effort to join our STEM Friday today. Christine, these were great questions. I really enjoyed the kids. And the questions that they did ask, I was like, wow, I never thought of that myself. So, but now this is our first time having her here. And usually we have people on rotation. So if you care to come back again and speak to the kids, about 6-7 months from now, we'll do this all over again.
That would be great. Yeah, I would love to do that. I really appreciate you guys having me. It was a great time. You all asked wonderful questions. Really, you were making me think.
That's good.
Yeah, it's a good exercise.
Yes. All right, guys. You guys have a
Okay.
great weekend. Thank you, Christine. We'll be in touch, okay?
Great.
And,
Sounds good.
you take care. Thank you,
Okay,
guys.
thank you. Bye, everyone.
Bye-bye.
Bye.